Hello everyone, welcome back to another episode of the Evolutionary AI series. In today's episode we are going to talk about genetic algorithms. Now this video builds on top of two previous videos of this series. In the first video we've been talking about Darwin, in particularly how he was going from Cambridge to the Galapagos Island, and then later on how he designed his theory of evolution. Then 100 years later we talked about Watson, Crick, Franklin, once again in Cambridge, and the discovery of the structure of the DNA. Then we linked all these pieces together and we defined evolutionary methods. Now today we will talk about one particular subset of evolutionary methods, that is the subset of genetic algorithms. Now, as you know, I like to follow a sort of top-down approach. I start from a high-level content and then I go down into details. In today's video, we will be high level. The goal here is to build the intuition. But then in future videos, we will go deeper into multiple aspects of genetic algorithms. Okay, so let's start from here. So in this picture, I will try to give you an intuition of how genetic algorithms operate. So on the top left corner, as you can see, we have a legend where we represent the standard organisms with a brown circle, organism that we call mutated type A that have a green dot, and then organisms that we call mutated type B that have a red dot. So these three types of organisms, they all live in one environment, same environment. So the environment is represented with a box. In the step number 1, the box is going to be reddish pink, and this means that in this specific environment, the red organisms, mutation type B, are the ones that have more chances of surviving, okay? Then step number 2, something happens. So the environment changes. As you can see, we go from pink to green. So the environment, the box become greenish. And this means that is in this new environment, the organism that is mutation type A has more chances of surviving. Now from step 2 to step 3, something is going to change. As you can see, the organism with the red dot disappears. So in the step 3, you will not find the organism mutation type B, because this can happen because the environment is not going to change for a very long time. And what happens is that the mutation type B is just going to get extinct. It's going to disappear from the population. And what does it mean for an environment to change? Well, in the natural world, change can happen in different ways. For instance, a portion of a forest can disappear because of a fire, and therefore if there were animals that were using the trees for shelter or for food, now these animals are going to be in trouble, right? Those animals could be the organism type B in our example. All right, then, if we go from step 3 to step 4, now what's going to happen is that mutation type A start to spread in the environment. So the environment remains green, is particularly favorable for mutation type A, and this means that in step 5 we will see organisms of type A just spreading in the environment and taking over. Now mutation type A becomes the standard organism. As you can see, it's taken over the environment. You have a lot of green organisms, and now the standard organisms are in minority and the mutation type B is completely disappeared. So in a nutshell, this is how evolution works, and this is the mechanism that we are going to use in genetic algorithms. Okay, next. All right, so in this example we are going a little bit more into detail about what's happening. So once again, we start from our initial population of organisms. For the moment, you don't need to understand what is an organism. We will talk about this later. So let's keep this generic for now. So we have initial population of organisms. We have one green, one red, and a bunch of standard organisms. And now, the first thing that we need to do is to perform fitness evaluation. What does it mean, fitness evaluation? We have to assign to each one of these individuals a real number, a value that is called fitness, and represent how how good is this particular organism for this specific environment. So as I told you before, the green organism is very well suited for this green environment, and therefore it will end up to have the highest fitness value, 0.56 in this case. On the other hand, the red organism is not very well suited for this environment. It's going to have the lowest fitness value, fitness score in this case. Then we have a bunch of standard organisms, and they are going to have a score in between 0.10 up to 0.35. All right, now step number 2, we have to perform what is called selection. We have to decide which individuals are going to be chosen, selected to reproduce and generate a new set of individuals. We will see later how selection is performed. For the moment, you can see here that we are just ranking the individuals from the best, the one with the highest fitness value, to the worst. And then we're going to just take a subset of all these individuals. In this case, we're just going to take 5 of them. All right, then step number 3, we're going to perform a recombination of these 5 individuals that will give us a new set of 5 individuals. Now these 5 individuals, you will see in this image, that are generated by using a pair of organisms. Now we can use the same organism here. You can use hybrid parents. We can use parents that are both standard but very different, and so on. And this will give us a new set of individuals. And this represents our new population, right? So a new population is just obtained by taking these individuals and moving them back to the environment. Once they are back in the environment, now we can evaluate the fitness of the new population, and we can repeat this cycle. So this is just a loop. Now we will go back to evaluate the fitness of the new population. We are going to select the best candidates, and then we're going to recombine them and generate another generation of individuals, and so on. So every time that we have a new population, we talk about a new generation. All right, but what is in practice an individual? An individual is just a chromosome, and a chromosome is can be represented in different ways. We can go with a label representation where each entry in the chromosome is a gene. So each entry is a gene, and in the label representation, each gene is a letter. So we go from A, B, C, D, and so on. Each letter may represent a subset of a solution. So you may think of a solution to a particular problem like a set of pieces that can be composed together. So the label representation is going to basically give you the single component of the solutions that you can piece together in different ways. Then we have the integer representation, where we have natural numbers in this case. It can be used to represent genes. In the floating point representation, we have genes represented by real numbers. So real numbers, 0.01, 0.33, and so on. In the bits representation, we have just Boolean values, 0, 1, true and false, and so on, representing the genes of the chromosomes. So which representation you should pick? Well, it depends from the problem that you are trying to solve. So for some problems, using floating points would be more appropriate. For other problems, using integers would be more appropriate, and so on. So if our set of solutions is a neural network, then in that case, the weights of the neural networks that are real numbers will be the genes of our chromosome. So a neural network can be compressed into a chromosome, and every gene of the chromosome will be one weight, one connection of the neural network. Instead, if we are evolving images, then in that case, maybe an integer representation is more appropriate, because we can represent bytes like number between 0 and 255, and therefore they can be just integers. Or we can go down to the bit representation for the image. In that case, we can go to the single bit and we can encode the image with a bit representation. So it really depends which type of problem you're trying to solve, but we'll go more into details later on in the course. Okay, now there is a question. How are individuals selected? Well, we're going to use natural selection, right? We've been talking about this in the video about Darwin. But what does it mean to use natural selection? So we are running these algorithms on a machine, on a computer. How can we recreate the mechanism of natural selection on a machine? All right, let's see. Let's try to answer this question. All right, so we're seeing that every individual of the population is represented by a chromosome that I represented in this image with this color sequence of shapes here, of rectangles. Now what we do in generation n is to take the 10 individuals and we're going to rank them from best to worst. Right? So what does it mean best to worst? It means that individual number 1 will be the best individual, the one with the highest fitness score, while organism number 10 will be the one with the lowest fitness value, fitness score. Okay, so once we have created this list of best worst candidates, now we can select them. There are two selection mechanisms that we will see today. The first one is called truncated selection. This is very simple. We're going to just split the population into two parts. We will take the top k candidates and we'll select them, and then we're going to take the n minus k candidates, where n is the total number of organisms, and we are going to reject them, to discard them. So the best k candidates are going to be selected and they are going to be used to generate a new population of organisms. So first method of selection is called truncated selection. Second method is called roulette wheel selection. In roulette wheel selection, we are going to define a pie and we are going to assign slices of this pie to each individual in our population. Now the size of the slice is proportional to the fitness value. So individual number 1 is the best, is going to get the largest portion of pie slice in our cake. And then we will go basically down, 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 down to individual number 10, the last one that will have the smallest slice of pie of our cake. Then what we are going to do, we are going to spin the roulette wheel, and by chance the roulette wheel will stop one particular location. In this case, as you can see, by chance we stop location number 2, so we are going to select the second individual, and the second individual is going to be used to generate a new set of offsprings. Then we are going to rotate the roulette wheel again. We're going to select another candidate, and this candidate is going to be selected to generate another set of offsprings, and so on. Then we will group all these offsprings inside one population, and this will be the population at n+1. Okay, now I would like for you to take 1 minute. You can stop the video, take 1 minute, try to think what is the advantage of truncated selection, what is the disadvantage of truncated selection, and advantage and disadvantages of roulette wheel selection instead. All right, so truncated selection is very simple to implement, right? So in practice, you just have a sorted list. You're going to take the top k candidates in this list. Now, roulette wheel selection, on the other hand, is slightly more complicated to implement. Technically speaking, in Python, it's trivial. You just have one line of code. You can do a random sampling, a weighted random sampling, but still a little bit more complicated. Okay, truncated selection is also more effective. Sometimes you want to focus on the best solutions and you want to rapidly discard solutions that are obviously wrong. So in some problems, truncated selection may be better than roulette wheel selection. However, truncated selection also has a series of problems, right? Because imagine, okay, think about the royal families in Europe. So royal families in Europe for centuries, they have been marrying and dating with representatives of the same families. For centuries, this has been going on. Of course, this mating strategy is not ideal because the genetic pool is very limited. And this created all sorts of problems. We had offsprings with severe health conditions that were just the consequence of this mating strategy. Now, on the other hand, roulette wheel selection, we don't have such a problems, right? Because we are not completely discarding individuals that have lower fitness, right? So we can still randomly select some of them. But on the other hand, by chance, it could happen that some of the best individuals may also get discarded. So it really depends how you're going to set the weights of these slices in the pie, and it really depends how you implement it then in practice. There could be also hybrid solutions where you start from a truncated selection mechanism, vice versa. You start from a roulette wheel selection mechanism at the beginning of the training for the first generations, and this will help you to keep inside the genetic pool also some of the worst individuals, and then later on, as you approach the end of the training, you may switch to a truncated selection mechanisms. Once again, this is just exploration versus exploitation. It's a mechanism that we will see also in reinforcement learning. So, at the end of the day, here it really depends how you want to implement this. Sometimes roulette wheel selection will be the best choice. Sometimes truncated selection will be the best choice. For sure, roulette wheel selection is closer in spirit to the idea of natural selection. The best individuals sometimes can die, right? And the worst individuals sometimes can survive. So roulette wheel is for sure more similar to natural selection. Okay, next. I've been talking about how to select a pool of candidates, but now the question is, how do we create a new generation of individuals? So we have selected the elite, we typically call it the elite of the population. Now we have to generate new individuals. How are we going to do that? So one way of doing it is called point mutation. So this also happens in biological organisms, where mutation of the DNA can generate variation of the DNA itself, and this is just a mechanism that we can also use here in genetic algorithms. So we have a set of parents, and then we're going to have a set of children. If we are in the first encoding type, if we are going to use characters, we're going to randomly select one gene from the chromosome, and then we're going to randomly switch it to another letter. So from D we go to K. In integers we do the same. We select one gene. We're going to randomly switch this number with another one. From 5 we can go to 4, for instance. Now in floating point notation things are a little bit different. We still randomly select one location, but then what we typically do, we use a Gaussian distribution with mean equal to 0.45 in this particular case, and a predefined variance, and then we're going to sample a value from this Gaussian. Now, depending from the variance, we may be very close to the mean. In this case, 0.49 is the value that we sampled. And finally, in the bit representation, bit encoding, also in this case, we are going to sample one gene, and we simply switch it with the opposite. So if we sample a location where there is a 1, we're going to turn it into a 0. If we sample a 0, we're going to turn it into a 1. And this is point mutation. As you can see, you can generate one or more children, depending how many times you're going to apply the point mutation. Another mechanism is called crossover. In crossover, now we need 2 parents. So crossover requires more than one individual. What we do, we randomly pick a slicing point, and then we are going to take, let's say, the first portion of the first parent, and we're going to assign it to the first portion of the second child. Then we're going to pick the first portion of the second parent, we're going to assign it to the first portion of the second child. Second portion of the second parent will become second portion of the second child. second portion of the first parent will become second portion of the first child. So visually, you should understand that we are crossing portion of the chromosomes. This is why we call it crossover. All right, we can apply crossover over multiple segments of the chromosome. In this case, we pick 2 slicing points, and we are just going to repeat the same process. So we're going to take first chunk of the first parent, and this will become the first chunk of the second child, then tail of the first parent will become the tail of the second child. And then of course we can do the same for the second parent. Head will become first child head, second parent tail will become first child tail. So once we have applied mutation, single point crossover, multi-point crossover, also in this case we have to pick the right transformations based on the problem at hand. So we have to define in advance which one of them we want to use. Some of them may be more appropriate for some cases, other for other cases. For instance, when we'll do near evolution, where we are going to use evolutionary AI genetic algorithms to evolve neural networks, we will see that we're going to use a floating point representation for the weights. In that case, a point mutation will be very effective. We can still use multi-point crossover and crossover to adapt the architecture of a neural network. We can consider one layer of a neural network and another layer of another neural network as different portion of the chromosomes. And then we can mix them together to generate the offsprings. All right, so that's all for today. We'll talk in the next video about how to practically use genetic algorithms to solve a variety of problems. But that's it for today. Thanks for watching, and I will see you to the next one.